Hello, mic check, mic testing, hello, mic testing, hello, 1 2 3 4 5 6, hello, mic testing, hello, mic check, hello, mic testing, hello, mic testing, light கேக்குது வாய்ஸ் லைட்டா இன்னும் கொஞ்சம் வேல்யூம் ஏத்து. ஹலோ மைக் செக் இன்னும் கொஞ்சம் ஏத்து. ஹலோ மைக் டெஸ்டிங், ஹலோ மைக் செக், ஹலோ மைக் டெஸ்டிங், ஹலோ மைக் ஓகே. மேடம்கிட்ட கட்டாக கேட்டு. ஹலோ மைக் செக் ஹலோ மைக் டெஸ்டிங். Hello, mic testing. Hello, 1 2 3 4 5 6. Hello, mic check. Mic testing. Hello, 1 2 3 4 5 6. Hello, mic testing. நல்லது சரியில்லையா? ஆஃப் பண்ணிட்டா? அதுவுமா? Next speaker. Okay.
Okay, so welcome back to the workshop. We are on day 9 now. So this is the last day of lectures. Hope you've been enjoying them so far. Just a quick reminder to the participants. So tomorrow will be your turn to give presentations. Hopefully you've been hopefully you made a start at making the presentations, and if you need any last minute feedback, please do approach us during the breaks today, right? So yes. Yes, so we the last session today will finish by 5:30. So we will have some time in the evening if you would like to meet with us and get some more feedback. Okay, so let's begin the first session of the day, and it will be the second talk by Professor Biplab Bose, and he'll be talking more about collective behavior of cells. So please go ahead.
Okay, thanks. Good morning, everyone. So if you remember yesterday's class, lecture, we just discussed broadly topics of collective behavior in cells and how phase transitions, concept of equilibrium processes and phase transition criticality, how we deal with those. So this is a particular example. This is a research problem that we were working for a couple of years. So in this case, we'll take a particular model which is inspired by a biological problem called planar cell polarity, and we try to understand that what type of phase transitions can appear in this particular model. The mathematical background, as I have actually discussed in the last lecture, so it will, I think, if you are not introduced to this subject earlier, it will be easier for you to follow. So before I start, this work is primarily was started by a MTech student, Kamleshwar. Then it was continued by Nirupam, another MTech student, and Himakar, who was a BTech student. So this is a development biology meeting, so obviously most of us we know that metazoan like us, we are made up of multiple cell types. So the issue of how diversity originates, it's a long-standing riddle, and we have lots of theories, ideas, concepts, and information also, right? Experimental data also. But at the same time, one idea that you have to keep in mind, that a cell which we start learning in school days as a like a sphere in which everything is in hodgepodge, right? All those things are there, like a symmetric one. That's not true. Almost every cell has some sort of polarity. So particularly epithelial cells which are on our surfaces, either outside or inside our body, right? Or suppose in blood vessels. So all these epithelial cells has a distinct polarity. By polarity, what we mean that there is some sort of asymmetry in terms of structure as well as function, both of them, right? It is not just a structural thing only. And when we say structure, mean not just a structure that you see on the cell membrane outside as a whole, but inside also. So particularly for epithelial cells, this type of polarization is very important, and which is very common, and we know is called apicobasal polarity, right? In that case, we have to specify direction, because cell has to adhere, because we know epithelial cells are adherent cell, they adhere to each other as well as with the basement. So those molecules which you have to express for this adheration, right, it has to be expressed on particular side of the cell. You cannot express across the whole cell everywhere. You are wasting energy and material for them, right? At the same time, for example, suppose this if you imagine these as a villi cells in your intestine, then if this side is the gut side, so then there must be extension, finger-like extension and protrusion through which the absorption will happen. Now absorption will not happen just like magic, so there must be machinery to uptake the nutrient. So all those machinery, subcellular machinery, should also be more here rather than on this side, right? Similarly, if this epithelial cell has to secrete something, then you will see the Golgi complex network will be more on the other side rather than this side. So this type of polarity is quite common in epithelial cell, and this is called apicobasal polarity. But I will not talk about that. I will talk about planar cell polarity. So apicobasal polarity, if you consider this as a z-axis and this is the x-axis, then you have another axis on the other side, because it's a three-dimensional object. So if you take a plane which is actually xy plane in this diagram, so it's a plane which is orthogonal to this apicobasal direction, cells can have polarity, that is an asymmetry in that direction also, right? And that is called planar cell polarity, because it is along the plane of these cells. And one interesting thing is that that will not appear only in one cell, but this is a coordinated or correlated behavior. So it is a collective behavior that will happen across thousands of cells, right? Now, this planar cell polarity is actually important for many processes, although most of the studies are done, molecular level studies are done extensively in Drosophila, from where the key genes has been discovered, and then through homology finding and extension of those in human and other mammalian system, we also see it now. For example, in the development of our kidney structure, there is involvement of planar cell polarity. Across all mammalian organism species, you will find the hair formation has some sort of planar cell polarity involvement. Now, we have to remember one thing, problem is that most of these events happen very early at the developmental stage. That's why their detection is also very difficult, right? In other cases, for example, in the eye of Drosophila, how the cells will get arranged. In your inner ear, how the cells will grow in a particular direction in a slanted way. All these things are controlled by planar cell polarity. Now, as I said, the most of the studies and iconic studies actually has been done in Drosophila. So, particularly in Drosophila wing. In fact, if you are now coming from experimental biology, even if you come from experimental biology, not developmental biology, if you read those papers and if you look into their experiment, this is an amazing experiment, mind-boggling in a way, how they implement them, right? How they create a mutant organism and then they study them, right? It's amazing experiments. And you really, honestly speaking, if you have any experience in experimental biology, you see this experiment, you will say that you need a courage to actually design and implement those experiments, right? It's not like, you know, every day run-of-the-mill experiment. Without training, you cannot do actually like that. So, so what happening here in Drosophila, just like we have here, Drosophila has bristles, right? And particularly in case of these wings, we call them trichome. They are hair-like, they are not hair in that sense. By the way, if you will notice your hair in a localized region of your body also, you will find this hair has a particular direction, right? Hairs in a particular region are all aligned in a particular direction, right? The story starts from there actually. In case of Drosophila wing, this hair-like structure, the trichomes, they are aligned in a particular direction. So if I consider this side of my wing as a proximal and this side is the distal one, so they will be on this direction. From proximal to distal, they are bent like this. This must have some sort of aerodynamic advantage or some other thing which really we do not know much. But it is not only slanted, but you have an interesting thing here is that they do not originate. See, if this is one single epithelial cell and one blade of this wing is suppose made up of around roughly 25 to 30,000 this epithelial cell. So this bristle could have come anywhere, right? It could have anywhere and then bent on the direction. It could have been, right? But that doesn't happen. It happens only in this one side, which is, we can say that if I consider this hexagonal thing on this distal side it starts. And obviously you can imagine this must not be a magic, and this must not be a very, from our experience in biological system, this must not be a simple one-step process. Multiple things happens before actually the location from where, if you say there is a seed position from where the bristle will start, multiple other events happen before that, and which actually decide the spot where it will start. One of the key events is obviously how cytoskeletal organization breaks and new structures are formed. But much before that to happen, one key event happened, and that key event is the main story of planar cell polarity in case of Drosophila wing. What is that key event? I have shown this in the figure itself, but let us look into further. So this is the real image. You can see the bristles are coming from one side, and thankfully in reality also most of the epithelial cell in this case are hexagonal, right? I mean, when I will create a model, I do not need to have to imagine and make an abstraction. They are hexagonal. They are actually hexagonal largely, though there are ifs and but. I will come to that. So as I am saying that before the position from where this bristle will start, before the asymmetric organization of cytoskeletal thing happen, something must happen, and that something must happen is connected to what we call now core PCP pathway. Actually, this was discovered first through mutational analysis, and then subsequently the molecules of this pathway has been discovered in human and other cases also, right? So they are very conserved, evolutionary very conserved. So what happened? What is that key incident that has to happen before, so that we can say it is a it is polarized. So what will happen? As I am saying, it is a collective behavior. So in thousands of all these cells simultaneously, what will happen? Two type of protein complexes will segregate. What do I mean by protein complexes? One is Van Gogh and Prickle. If you are not from developmental biology area, actually these names are very interesting. Why they call it Van Gogh? Because if you mutate this one in a swirling pattern, this pattern breaks down and you have swirling pattern of hair comes. Right? Quite imaginative, isn't it? I mean, it's not boring, remember things you have to remember. This is very easy to remember name, right? So it's a Van Gogh and its partner is Prickle. There are other molecules also involved in this complex formation. We are just skipping it so that we can keep focus on the whole, the simplified idea what is happening. So I am representing them by red color. They get segregated in the proximal edge of the cell. Now this is tricky to do experimentally. Means, see, if you do something like this, if you have to do a design experiment, the obvious thing you will say, okay, why don't I tag it with a fluorescent probe, or I use antibody against Bang to see where it is accumulated, right? But the problem is you will not be able to see whether it is accumulated here or accumulated here. So they have this classic mosaic experiment where you have a wild type cell mixed with what we say the mutated cell where the Bang is not there, so you can differentiate whether they have these red fellows have accumulated here or accumulated here. So from these beautiful experiments, what they have proved that this Bang and Bang is a transmembrane protein and Prickle is its cytoplasmic partner. They accumulate in this region, whereas by blue what I show them, which accumulate the protein complex which accumulate on the distal edges of these cells is Frizzle, Disheveled and Diogo. Fine. So this is the key event. When the protein expression happens, early stage of larval development, if you look into these cells, you will see that actually these proteins are largely diffused throughout these cells. But then slowly they segregate, right? And they don't only segregate in one cell, they segregate across the whole layer of cells collectively. And once they have segregated correctly in this fashion, then only you can expect that cytoskeletal arrangements will happen properly, polymerization, depolymerization happen in a particular way, and then from these positions a seed structure will form from which the bristles will come up. So if you can disturb this segregation, subsequent processes does not happen, right? And if they happen in a wrong way. You get different patterns, right? So what's going on? So remember, this is a mind-boggling complicated process. Yeah?
Proximal and distal directions.
Sorry?
Proximal and distal directions.
This direction is proximal, this direction is distal.
This is defined with reference to what?
My wing is expanding like this from my thorax body.
Right.
So this is my proximal side, this is my distal side.
So these bristles are found in the wing?
Yeah,
on the wing or on the
wing.
body?
These are on the wing.
Oh, I see.
They have the body, in body also they have, and that also planar cell polarity. From the, for example, dorsal side they have sensory bristles. Those are also a part of the planar cell polarity. So, but we are discussing this one for the wing, and we have to remember that actually although we will say these are all planar cell polarity, the exact mechanism may vary even in the same organism. Largely same molecules may be there, right? So we should focus, if you say PCP, you have to be very careful which PCP you are talking of. This is the most extensively studied one, PCP of Drosophila wing. And the PCP of in their compound I, there is a PCP, but that system is different. So we should be very careful about that. So now one thing I will ask you to remember. See, as I was saying in the last lecture that we want to create toy models and we want to get rid of lots of clutter, right? Before even if you go to understand and make toy model in this type of case, we have to remember that what we are talking, which the essential distal thing that I will pick up as a logic of my model is coming from which is largely consensus among the community, largely consensus, right? So this idea that there is a core signaling module involving these molecules is largely consensus. There are many other processes which are happening simultaneously, for example, and we will not discuss or capture them because these are still in a fluid situation as in our understanding, is that, for example, these cells, they are hexagonal, yes, your image you can see, but it's not like that. When in the larval stage, when this wings is forming, obviously if you remember yesterday's lecture, the wing is not formed in one go, right? It step by step increase, expand and then get thinner, right? So during this, actually what happens, very interesting thing happens, what we call neighbor exchange. One cell exchange its neighbor, right? If somebody, a cell A was neighbor of cell B, cell C was neighbor of cell B. Now after some time, they push and pull each other, change the interactions, and C becomes neighbor of A, B is not anymore neighbor, right? So this neighbor exchange happens, and when doing this, these cells do not retain this hexagonal structure. They sometimes become pentagon, octagon, something like that. So there is some sort of fluidity, it happens, right? But eventually, if you consider a steady state behavior, the steady state is definitely this hexagon. We have to remember that. There are claims of movement of cells, right? As well as at the same time, we have to remember that, see, this process does not happen just one go like this. It is not happening like that, that I have a plane of cells in which everything is happening together, although in our model we will consider that. We have to remember these are happening in stages. Interestingly, suppose up to this the wing has formed, planar cell polarity happens. And then when it extends, again it gets disturbed, again it happens, right? So there are lots of things going on, but we are focusing on only the essential consensus part. And one important point I just missed, I thought I will tell you, if you are coming from other biological this field, yeah. Is this why it is happening?
Yeah, like
Yes, so the whole issue is when this wing development happens, there is one predominant thing which is evolving now as a understanding is that, you know, you have mechanical forces right there. So those forces develops as I am unfurling my wing, if you think that way. It's not that unfurling only, you have directional cell division also, right? So in that time, actually you have a particular type of mechanical stress generate, and that deform your cell-cell interactions. As well as your cytoskeletal structure is continuously going through deformations, modifications, right? That gives a fluidity in the system.
Yeah, like because if new neighbors are established, that means they have to establish their connections also, right?
Yeah. Everything breaks, everything open.
Yeah, it will be an expensive process.
Yeah, it is an expensive process, definitely.
Okay, thank you.
Right? In general, if you forget about anything, any order formation is an expensive process. You have to do some work. Somebody has to do some work from disorder to order somehow, right? You can think that way also, right? So now, one thing I thought I will tell you, because it's not just that, see, we repeatedly will tell that PCP is a part of phenomena in epithelial cell, but that's not true. In the EMT community, actually now one important consensus is coming up. Actually, mesenchymal cell also show planar cell polarity because when they move in group particularly, right? Now we have evidences in clinical things also that mesenchymal cells migrate collectively, cancerous cells migrate as a mesenchymal form collectively, and then actually when the direction they are moving, the cytoskeletal arrangement has a polarity, which direction they have to move, right? So that's also a planar cell polarity, also in mesenchymal cell. Neuronal cell also shows to some extent like that in the early development. So now, so as I said, we will focus only in this discussion only on this core pathway, right? Living around all these clutters. So in the core pathway, what happens is that very interesting. What over the years, almost 30-40 years, data shows that the core pathway, as I said, involved in this molecule, they have very interesting behavior. One is cell non-autonomous, right? The neighboring cell has interaction. How that interaction happens, that's very interesting. So in the proximal site, distal site of a cell, this is one cell, this distal site is this one where you have frizzled, right? Frizzled is a transmembrane protein which is has a WNT pathway molecule, and it has a partner in the cytoplasm called disabled and Diego. They make a complex, and there is a transmembrane protein which is called flamingo here. And interestingly, flamingo will also accumulate on the other side of this cell, which is the distal side of this second cell, and they have homophilic interaction. This is essential. If you disturb that, the whole process will get disturbed. But what happens is that in this case, frizzled also has a, I think it is called wingless, right? WG. Wingless is the ligand for this one, is a WNT molecule. But in this case, it is not interacting with wingless. What it does that it will interact as a whole. This complex will interact with the Wang and prickle complex on the other side of the neighbor, right? And these interactions are heterophilic, but in presence of this whole thing, this is stabilized. this is very stable, and it is so stable that actually very stable punctures are formed here, right? You almost like if you do those fluorescence studies, you will see the diffusivity is very low. They are not moving. They are very stable punctures are formed. But the most, so that means what I can say, in the neighboring cell, complex type A and complex type B are liking each other, right? They favor each other. But the interesting thing is inside the cell, in an individual cell, intracellularly, right, they actually push each other somehow. This part we do not know clearly how exactly that happens. Some of the clues we have, but experimental evidence grossly shows that it is happening. For example, you have actually some sort of circulatory motion through which these molecules get segregated, that get affected if you modify some of these molecules, mutate them. Then you have frizzled, which will phosphorylate Vang, right? And if you do that, so then this process will get disturbed. At the same time, you have disabled and Diego, they compete for prickle. So if prickle bind to any of them, then this complexation will not form, right? And these edges will not form. So that means inside the cell there must be some processes by which they get segregated. So if I boil down this reduced form, the logic comes for the thing is that there are two types of local interaction in this system. Neighbor to neighbor, the opposite protein complexes, the frizzle and the Vang, if I consider them as different protein complexes, they love each other, they interact favorably with each other and stabilize, whereas inside the cell they push them each other. Fine? Now you can think about, this is what we call the local interaction in PCP model. And obviously, if you remember yesterday's lecture, the collective behavior's hallmark is the whole behavior arise because of the local interaction, right? And if you remember in the last lecture also, I was saying that does not mean there is no global cue. The difference is that there is no global authority. There is no global pattern in which you are putting things, right? But local interactions is working, but at the same time you need some global global cue. For example, particularly for this case, so if you consider this one, that okay, this picture will be much better actually, I think. So if you think that there is a vector I can draw, right? So from this red to this blue. So the vectorial arrangement is in this direction, proximal to distal. But it's a hexagon, right? If you only think of local interaction, then this vector can have 6 directions. Logically, you don't need math to do that, right? Because anyway, that will give me this type of segregation. Fine. So, but that is not happening. Out of these 6 possibilities, which are actually identical in a way, if you forget about which is proximal, which is distal, right? So, this is happening, only one vector is preferred. So, that means there must be something, some global cue to tell, see, this is the distal side. You have to go, you have to point on that direction. Now, this is very debatable. What is that global cue? Initially, if you look into the old paper, the whole idea was there was something called 4-jointed and Dassus pathway, right? Actually, Dassus FAC pathway. So, this Dassus is a transmembrane protein. Its partner is FAT, which is also a transmembrane protein. But they interact and stabilize at the border of cells. But that process is controlled by 4-jointed, which is a Golgi apparatus molecule. And the interesting thing, very early in the development studies of this type of system, it was observed that there is a gradient in expression of 4-jointed. For example, 4-jointed will be higher on this distal side, whereas Dassus expression will be higher on this side, right? So there are mathematical models also where people use this idea to show that this helps in segregation of this molecule and eventually giving that pattern. But then it turned out that actually their presence is important. I mean, these molecules are required for the process, but the gradient is not. If you disturb the gradient also, right, then still also you get the pattern, right? So that means this gradient is not working as a, although the gradient exists, it may have some other utility, but it does not, is not the global cue. Then people thought, because there are some experiments where we have seen the WNT molecules, the soluble and diffuse one, has a gradient, but it turned out that actually if you remove those WNT molecule itself, nothing happens. Your cells still get polarized and your hair comes. Now one thing is evolving, which we still do not know exactly, is that possibly this mechanical stress that originate right during the development of the window, that may be giving the global cue, and that makes sense because that is also already controlling the directional cell divisions, right? So possibly that is a global cue, but we cannot vouch on that till now unless a consensus develop. So we have to remember for this talk that we will focus on this global interaction, local interaction between two neighboring cells through these opposite molecular complexes, and inside the cell these complexes push each other away, and there must be some sort of global. I will start without the global and we will see the behavior, and then we will go into why the global is there, why possibly we are failing to detect the global. All these questions we will try to address. So there are lots of beautiful mathematical models in that by even by famous people actually, right? So remember these are different, starting from ODE based model where you use bifurcation theory to then to create the asymmetry, right? Then this PDE based model, then you have cellular automata type model. Every type of models are there, large number of them, and most of the time you have to remember these were done beautifully to justify the mutational experiment data. You mutate one thing and you see this type of pattern come, so you create a mathematical model based upon that signaling pathway to see that it is true or not, right? But one important thing that we want to touch upon is that, see, as I said, initially the molecules remain largely diffused and then they segregate, right, and make this thing, and that happens collectively across all these thousands of cells, and we know this involves primarily the local interaction as well as some sort of unknown global interaction. So definitely it falls into the bag of collective phenomena, and paper after paper, if you read on planar cell polarity, whether research paper or a review article, they will say that is a collective phenomena, and many of them will move one step forward to say this is actually symmetry breaking. Yes, in the concept of symmetry breaking, definitely this is symmetry breaking, and so it is almost like a phase transition, and many of them will also say it's like a ferromagnetic behavior, it is a paramagnetic state almost to a ferromagnetic one. Now the problem, as I was telling in the last lecture also, that to experimentally check whether this is actually a phase transition, and if it is a phase transition, what type of phase transition, where is the criticality, which parameter is possibly the critical parameter, we really do not have anything, right? So what we thought, why don't we create a very simple toy model which will not be trying to justify what is happening in planar cell polarity. No, we are not trying to replicate planar cell polarity in wing or something. What we want to say that, okay, see, if you distill out all this information where the molecular and cell biologists are giving us, and if I make into a rule, just like when you create a, what do you say, logic models for cell signaling, you essentially convert into 1-0 logic and or logic gates, right? So you are distilling the whole process into certain set of logic in Boolean network, right? So similarly here, if I just get the logics on which we have the consensus and create a system which will be obviously a mathematical object which will implement those logic, and then I see what type of behavior can come out of that logic, right? What is possible thing. So in this case, what we have a consensus? The consensus is there is local interaction, and local interaction of two types. Neighboring cells, neighboring cell will have two complexes which have favorable interaction in the border, whereas inside the cell these two complexes should segregate out, right? This is the minimum one. So this is the basic logic, and that is implemented by all those molecules, but essentially the logic is this one. And then what we have is that the second one, which is the global one. So the first model that we will discuss only deal with the local one. So we use this model, I will explain this. What we have an array of hexagonal cell. It is a triangular lattice because each cell has 6 neighbors, right? And thankfully largely in reality also these cells are hexagons, so we do not face much dispute in this case. And what we represent, see, we represent this, you remember there are two types of molecular complexes, right? One is frizzled and associated partner, and another one is a Van Gogh associated partner. So forget about those names for the time being, you can consider them complex 1, complex 2, like that. And for simplicity, let us color code them, right, red and blue, and they segregate. And for mathematical representation of that, not for any other reason, because just I have to write a logic equation, I will consider one of them, this red one or the protein complex 2 as +1 spin, whereas the protein complex 1 as -1 spin. So if you remember in the last lecture I was telling that the Hamiltonian in your Ising model, you can calculate from quantum mechanical calculations. There is not, many people have done that, right? But actually you should focus more on the logic in the Hamiltonian, what is getting represented, then you can extend it in so-called non-physical thing. So-called non-physical thing mean which is not classical mechanics or quantum mechanics things are coming, where the standard energy for force things are not matching, right? We do not know that. Right? So in that sense here we are considering this rate protein complex 2 as a spin plus, this minus. You can swap them, it doesn't affect, right? So then what we have, see what we want is that ideally in the equilibrium state, right, these protein complexes inside a cell should segregate. Yes, in B if you see they are segregated on the opposite side, right? Then the second important thing is that between the neighbor, this blue and red should be on the same edge, right? Because in the between the neighbor they like the opposite thing. So here I have blue in this Q cell, B also has red, this R cell has blue, this is red, and this one is empty, you can consider it 0 spin, it also has 0 spin, so matching. This is 0, this is 0, this is blue, this is red, this is blue, this is red. This is the ideal condition, this is the equilibrium that you see in your data, right? Now this thing can happen in other direction also, because if I don't have any definition of proximal and distal, that means I do not have any global flag, then it can happen in any 6 possible direction. So for example, in this case you see A, it has this segregation, polarization, but it is in this direction, not in this direction like B. So here also red, blue, 0, 0, red, blue, this matching, and inside also the segregation has happened, right? So you have to write a energy function, the Hamiltonian, which will favor this type of alignments, right? This type of segregation and alignment, fine. So the Hamiltonian that we will use is very simple. It will capture that logic. So if you consider the first, that +1 in one cell will interact favorably with -1 in another cell, right, the 2 spin. So you are multiplying these 2 spin and summing over all pair of neighbor, and you are multiplying with the interaction constant m1, where m1 if you make it greater than 0, so it will be negative, right, sorry, positive. So then when this spin and this spin are opposite, then this whole term is negative, so it is favorable one, right, that's the logic, fine. Now for the inside cell intracellular thing, you want that this red fellow should not be along this blue one, rather this blue should stay with the blue, that is a favorable one, this red should come here. So what you are doing, this is one spin, this is another spin, you can have 6 pairs here, and you are multiplying their spin values, and in this case you make m2 negative. Then what will happen, this type of segregation where the blue and blue are neighbor, red and red are neighbor, will be actually favorable one, will have a negative value, large negative value, right? Now you can tell me what this m1 and m2 represent. In case of your Ising model, you have J, which is representing the interaction energy in that term. We consider this Hamiltonian and all these things dimensionless, because we do not have a definition of energy in this case. We do not have any definition of temperature either, right? So what we do, we consider them dimensionless quantity, but what is the physical meaning of this constant? This physical meaning is, remember, just like any other equilibrium system, what is happening here, I am considering that there is thermal fluctuation or any other fluctuation you can think of, which is maybe thermal origin or some other origin, whatever it is. So there is fluctuation, and the fluctuation will try to distribute these spins across uniformly, right? That's what it should try to tend, right? But at the same time, these two of local interaction that's between 2 cells and within these cells will try to oppose them. So this m1, m2 is a relative strength of these local rules with respect to whatever randomization is happening inside the cell. You can imagine that, right? So these we can take different values of m1, m2 and then do the study. For most of the study, we usually consider them as the same value, so that I don't have to deal with lots of simulation. Fine. So now comes simulation. How we will do is standard Monte Carlo using Metropolis algorithm. What we do, we randomly pick one of these cells, right? And then suppose I have picked up this cell, and in this cell I'll randomly pick any one pair of spins or the protein complexes, right? And then what I will do, I'll swap their position. That is representing that they are getting changed because of the randomness, right? And once we swap, I calculate what is the change in energy because of the swap, and then I accept the swap with the probability p using the standard Metropolis logic. The minimum of either it is 1 or e to the power delta h, and you must have been noticing here we do not have temperature here, right? Because everything is scaled as a unitless in that sense. Fine. So now one important thing we have to remember, what do you want to study? So if you use Metropolis algorithm, obviously it will eventually lead to a equilibrium configurations, right? And you can sample from that equilibrium configuration and measure lots of property. But before we define those properties, a few things we have to define. See, what is the goal? The goal is all cells, suppose I have 40,000 cells, all cells should get aligned in a particular way. What do I mean by aligned in a particular way? That means inside the cell, this red and blue, these two spins or these protein complexes should segregate exactly with the way we see in biology, in the opposite direction, and at the same time, this neighbor-neighbor interaction should be correct the way we see in biology, right? So in this example, for example, we say this B, obviously it is polarized, you can easily see there is a segregation, right? But it is also aligned because you say B has inside the cell and across the neighbor, it has the right orientation. C is also like that, if you pay attention. So B and C are neighbor. So I will say B and C make a cluster of two aligned cells. In general, when I will say cluster, I will say they are aligned cell actually. So B and C is a cluster of aligned cell. I want the whole system to be one single cluster actually, right? And whereas A is aligned, but it does not have any neighbor which is aligned properly. So this A is a cluster of size 1, because cluster size can be minimum, can be 1, right? So that's all, fine. So now you can define other properties based upon these rules, right? Okay. So what do we see? As I said, to keep life simple, you start with both the M's as same, only the sign is different. So you see like this, is that as you keep on increasing M for different lattice system, the fraction of cell aligned means they have this segregation as well as the neighbor interaction is correct, as I said. It eventually saturates, and it's a beautiful sigmoid function which beautifully fits with a what we say Hill function, and you will expect a Hill function type of behavior, because if you remember, this local interaction is neighbor-neighbor interaction, so it's a cooperative phenomena, right? So cooperative phenomena are very nicely captured by Hill-like function. So in this case, it is actually capturing, but what interesting thing is that it actually does not depend much on our lattice sizes. Usually, if I remember correctly, Drosophila wing have around slightly less than 30,000 cells. So anyway, 256 is good enough size, right? And we have periodic boundary condition also. So therefore, so far so good, cells can get aligned because of this local interaction. But that's not much interesting to see. This is not phase transition, obviously. This is a smooth sigmoid behavior. So what we do now, we try to look images, right? So this is one single snapshot. When M's, M1 or M2, they are very low, 1, you can see there are some dots. These dots are colored blue, red, black based upon the size of the cluster. So red means the largest cluster, blue means the second largest cluster, black means the rest of the cluster, and white means there is no cluster. Fine. So remember, our cluster could be size 1 also. So when you have 1, you have dispersed small cluster here and there. If you increase 2, you can see some clusters are forming which are bigger, right? And I can easily see this fellow is close to this one, so possibly if I increase M slightly more, they will get joined, and then I have a huge cluster, and that's what exactly happened. Here 1 to 2 I am jumping, whereas I am from here 2 to 2.2, a slight increase, and the whole lattice get filled by one single cluster, right? Yeah. This is a triangular lattice, but we are plotting in this way. So you have this zigzag, right? But when you make this image, you do not see that. Colors means red means the largest cluster. So this cluster form here, and it wrapped around because you have periodic boundary condition. So that's the largest cluster. Second largest cluster is the blue one. The green one is the third largest cluster. Rest of the cluster are black, right? So looking at this immediately, if you are working on this type of model, you will recognize that, oh, there is a phase transition, right? This is typical geometric phase transition, what you see in percolation that I was discussing in the last lecture, right? So that means possibly here we are seeing a percolation transition. So if I see there is a percolation transition, then I have to check out what is the behavior of this percolation transition. And as I was saying that we define nowadays percolation strength, which is the order parameter, as size of the largest cluster divided by L into L. This, remember, it is valid only when we are working in terms of numerically, where clusters system is finite, because otherwise it doesn't have any meaning. Infinite size cluster doesn't have meaning in this case. So it is a finite size largest cluster divided by the system size. And you can see here we have a beautiful threshold behavior. Somewhere here I have a critical point for M1 and M2, after which below that the largest cluster size is almost 0, and then it becomes very big, right? It increases. And ideally you want that everything should be here, right? So that means based upon M1 and M2's value, we have some sort of percolation transition, right? So you have to characterize it further. The second thing that you obviously will do is to look into average cluster size. So average cluster size will actually behave like correlation. You can calculate correlation length in this case also. It will behave diverge like that only. So it is much easier to understand. So what you have, average cluster size is you are actually calculating the average size of those clusters which is not the biggest one. Because if you remember from the last lecture that the percolation theory develops where the percolation happened, and the cluster that is percolating is of infinite size, because the system is at thermodynamic limit, right? So when you do for finite system calculation also, you actually remove that finite largest cluster and only take those clusters which are not the largest one. So what you can see here is a very beautifully, you can explain what is happening. When M1 and M2 are less, very small, below some sort of threshold here, what is happening? Small clusters start happens, right? And as I approach from lower side towards this critical point, these small clusters start forming, joining, right? And the larger one form. So even if you remove the largest cluster, the average size of the clusters in the system will increase, right? And it diverge like this, so it increases, and then somewhere here there is a critical threshold where all these large clusters, they fuse with each other, making a system-wide cluster, right? So that's why here I should have a divergence, right? Because a huge cluster is formed, right? And I am not considering them here. Now if I move away from them, then what will happen? The system has a system-wide cluster. Here and there few cells are isolated, they are alone, they are loner, they may be forming cluster of size 1 or 2, something like that. So the average cluster size starts decaying, right? So the most interesting part is happening here. Here actually when I am reaching the critical threshold, some cells, they are organizing themselves initially, small cluster, but that is based upon local interaction only. But as I reach towards the critical threshold, this local interaction spread and I get a global correlation. So I get a global order, right? I am here in the cell, I have got aligned with my friends here, you are far away there, but when we are in this, close to this critical threshold, how we are getting aligned is affecting you, fine? So a global order origin comes. So that is typical of any phase transition. That's I'm trying to explain if you are not familiar with that. So this diagram shows that very nicely.
Can we say that after the critical threshold,
Yeah.
so the strength of the interaction is so much that a particular cell in a cluster will actually leave the cluster and move towards the other cell, so that it's effectively decreasing?
No, nobody is moving. What is happening here, if you are here in this region, see, you can see size of the largest cluster somewhere, suppose here, is very big, right? Fine, almost the thing. So what about the other? There must be other cells must be there, fine? So they are making small cluster. As you move further, what is happening? Most of the cells are becoming part of the largest cluster, but few loners are here and there. Now as I move further and further from the critical point, those number of loners decrease further and further. So you are calculating average size based upon that, because you remove the largest cluster from your average. So that means now average cluster size will drop. Got it? Okay. So if you remember, we were discussing in the last lecture, and we were debating also, that one way is actually to focus on that if you make a different model for different system for phase transition, we always want to understand what is happening near the critical point. because obviously because it is difficult to understand. At the same time, we want to actually categorize our real system or model system in different categories, right? So that's why the concept of universality came, that we discussed in the last lecture. And now we define multiple model belonging to the same group or same class or universality class based upon some sort of scaling exponent, right? For example, I can feed data here, so this will be a power law thing. So I must have an exponent to that, right? So one can actually, I have discussed this thing. For example, the percolation strength will have beta as the exponent and gamma for the average cluster size. But what I have not discussed this one, just briefly to remind you if you do not know. See, these things has been derived considering, it is derived, it is not assumption or something like that, are considering the system of infinite size, right? But you are not dealing with an infinite system. Even if you consider periodic boundary condition, it is finite system in a way. So then what happens is that you can imagine what is happening. See, in case of a infinite system, near the critical threshold, the size of the cluster is becoming very large, right? So if you can see that the correlation length means, suppose I calculate the average length of correlation between different cells, whether they are aligned in the same way or not, is becoming very large near the critical point, because distant cell will have the correlation in their behavior, fine. Now, if your correlation length itself is infinite, but your system of simulation is finite, then you are in trouble, right? You are truncating, right? Even if you don't consider it as an infinite correlation length, if you consider a very large correlation length also, and if your lattice is not of that size, then you are again in trouble, you are truncating actually, right? So that means if you are in a region and you have designed a system where the correlation length is bigger than the linear length of your system, then you are in a trouble because you are truncating the events, right? So in that case, it can be shown to some extent analytically, and then lots of guess and assumption is that that actually in that the scaling then will boil down with respect to L, not with this reduce or control parameter, right? So it depends upon the not the correlation length but on the real length of your system. So there is a whole field of thing. I thought yesterday I will teach you some part of it, but anyway we skip it. So I can actually come from here to here. Again, I get a scaling relation, but now with respect to L, right? And this function, we really do not know what is the exact function. It's a function of how far I am from the critical point and the system size to the power of this exponent. What we can do data collapse or data, fit data in a sense data fitting to calculate this parameter, and I can get this function also, right? So that's how you do it, because this is an ideal case, this is your real systems case. So you have to fit your data, P and chi data, and you know the L, you know how far you are from PC in your data, and you can calculate beta, nu and gamma, fine. So there are many techniques. What we use is a simple sort of data fitting where actually they will feed the data to this equation to get this gamma, beta and nu. Now, but we have a problem. The problem is all these answers that have been derived based upon considering a classic 2D random percolation, where the control parameter is probability of occupancy of a grid, right? The probability with which you make the grid black. We don't have any such probability in our system, right? We have only one thing, is that M1 and M2, and we are considering M1 and M2 as the same in our simulation, right? So how should I go back here? The only way forward is with a leap of faith that if I consider fraction of cell aligned as a equivalent thing with respect to P, right? So what we do, Fa is the fraction of cell aligned for a particular value of M. How can I get it? If you remember, we have this plot. This is the fraction of cell aligned. So these are the M1, M2. So this is actually a probability term, right? Because from 0 to 1, it's a probability in a way, right? So I have done simulation with different values of M, so I know what was the fraction of cell aligned, right? So I use that fraction of cell aligned as a proxy of P, and we consider that, okay, there must be scaling relation will be valid, and anyway this is an unknown function, universal function that we have to get from the data. So what we do now, we replace this P and PC by fraction of cell aligned for P and the critical one, this is also unknown, we have to get from the data fitting. But in terms of action of cell aligned. So what we do the data collapse now. So what we do, we rearrange the data. If you remember in this plot, this axis is M, but now I change that to axis to FA, fraction of cell aligned. So now the data looks like this, and you can easily see the effect of system size, which is visible now in these two plots, which was not visible earlier so clearly. So now what we do, we feed this data to those scaling equation, and we get these values of what we say, nu, beta by nu, and gamma. And this is the standard textbook values of 2D percolation, which are considered as standard. And if you consider our model's values, these are within the error, they are very close to each other. So from this, we can claim that that means this so-called spin-based model inspired by planar cell polarity, where we have only considered local cell, local interactions only inside the cell and the neighboring cell, belongs to the 2D random percolation class, and that these things may not be very, in terms of clinically or biologically so interesting, but if you think from a physical science perspective, that you are starting with a model which is much more complicated than simple 2D random percolation. 2D random percolation is nothing but you have a board in which you are throwing darts, and darts are hitting somewhere, you will get a hole. Is actually 2D percolation, right? That's so simple. But if you create this type of model, eventually it boils down to this 2D random percolation class, and that strengthens this philosophical belief that even though complicated systems has complicated rules possibly, but the underlying their structures are very simple, right? And possibly we repeatedly need to remind us that one, because otherwise it's very difficult to work with the complicated problems, right? I will skip these things, these are nothing much to entertain. Let us come here. So what we have discussed till now, two parts. One is I discussed the biological problem based upon we set up the model and we have shown the behavior. But remember, everything is based upon your local interaction, cell-cell interaction within cell interaction. So I wonder somebody could have asked, see in this case when I am saying alignment of cell, alignment could be in any 6 directions, right? So even when I say the whole system is aligned, it is in any 6 direction in my simulations also, right? I have no control over it, right? Now that doesn't happen in biology, and as I am arguing, you don't need to do any simulation or experiment to check that. Your common sense say there must be something to break this 6-fold symmetry, right? That I have to align in one particular direction. So what is that? And that's why people have tried to find a global Q. That's why they are so desperate to find a global Q, because you cannot rule it out. So, and as I said, we don't have a consensus. Every time a new contender comes, somebody else after a few years proves that it is not really working as a global Q. So what will happen if I include a global Q? That's the first question. The second question is that why we cannot find the global Q, right? These questions obviously box everybody who is working either experimentally or theoretically in this field. So what we do a simple thing that my system is very easy to manipulate. I don't have to write lots of PD or something. I can simply add another layer of which will give me the global Q, fine? So what I do now, I decide this side is proximal, this side is distal, something like that. To rather confusing distal proximal, I say this is the up direction, easy to understand, right? And I want all cells to align in this up direction. For example, in this case, X and Y, they are aligned in the up direction because see, they have internal segregation as well external neighbor interactions are also fine. Whereas this fellow Q, it is aligned, but it is not in the up direction, that I don't want. So I will not consider it anymore as aligned. Although it has internal segregation in the proper way, I will not consider that anymore. I have to consider some sort of external thing which will actually tell that, see, this is the up direction. You can imagine many ways, some can be simple, some can be complex. The one simple way you can think about, what about a field, some sort of field. When you say there is a WNT molecule diffusing and creating a gradient, you are actually saying you have a molecular field, right? And that field is actually giving direction, fine. So you imagine some imaginary field is there. If you consider there is a mechanical stress also, that you can also represent like a field, right, which is decaying in. So imagine some sort of field is there and that interacts with the spins, fine. So what I want is that these positive spins, this red one, right, are preferably should go on this side, right. So that means there is a field somewhere, and when they move in this up direction, that will be favorable, fine. And these are regular structure hexagons, so you can actually calculate the displacement very easily, standard one, right? No big deal, school level geometry. So what we do, now we modify our Hamiltonian. This is the Hamiltonian we have seen earlier. Now I create another term, del G, which is because of the interaction with the external field. So this is a constant, I will come later on. So suppose this is a spin at of the alpha cell at A position, any of them you can consider A, and this is for another spin in the same cell which is in B position, and you are swapping their position. In your simulation, you are swapping their position, right? So that means if I am here, I am going up, this fellow was up, it is coming down. So one is working along the field, one is going against the field. So going against the field must have more energy cost, whereas if I going on the up direction, I should have less. So now that also depends upon what spin I have. So we consider, obviously these displacement must be same, right? But their directions should have a value. So if I go up, I consider them as what we say, positive, and if you go down, I consider them negative, and I consider M3 as a constant in this case. You can consider that as a field strength as a negative thing, fine. So what will happen in this case, this positive spin will favorably go in the up direction when M3 is a non-zero value, and the negative will preferably go down side, fine. So then everything is same. So what happens? Okay, this is what happened. Now remember, I have changed the definition of alignment here. Any 6 direction is not done anymore. Only in this up direction, if you are aligned, right? So we calculate the fraction of cell aligned. Now I cannot escape because I cannot consider M3 as same because there will be trouble. So we consider M1, M2 same, M3 as another value. So we scan through the whole thing, and we see there is a large area where in the state space where all cells will get aligned, right? Because this is 1, so you can imagine all this area where all cells in my system will get aligned in the right direction, fine. Now the question comes whether this is also a percolation. Yes, this is also percolation transition. You can see here this is happening, and nothing much interesting in terms of percolation, because you can easily see that if I vary M3 and if I keep M means this one constant, we still see this percolation strength, same percolation type behavior. We did all those universality classes, those nothing new, they are same. So I will not go in detail of those. In one thing just to remember percolation in principle does not require any interaction, right? If you remember the last class, right? We do not have this issue of interactions there, right, in the 2D percolation model. So here also you can see actually if you have M3, actually in absence of M0 also, even if you don't have local interaction, the system can percolate, and that's quite obvious. I mean, in this case you have to have the M3 very high. Obviously the threshold is becoming very high. Means essentially what I am saying that actually you can achieve this global alignment, simultaneous alignment of all cells even without local interaction. This is the riddle, right? This is a riddle, I will come back to this, and this is not a riddle in this field only. In almost every field of collective behavior, this riddle comes, that I can achieve the same structure by using some global authority, global Q. Then why the hell I have the local one, right? Because it costs, right, to have that. So this is happening, you have to keep in mind, I will come back to that. And obviously you have all other properties. As I said, I will not go in detail of this. Let us come back to what new we learned. If you talk of in terms of percolation in this particular second model, nothing new will come. But this can actually give us some clue about certain other questions. The first thing I will ask you to look into this, this seems very obvious, but we have to be careful about here. So this is a parameter state space, right? This is parameter, this is parameter. You can see such a huge parameter space is available to my model to achieve what is target it want to achieve, right? Now this is not settling for a biologist, because remember, in any biological system your parameter space which is suitable for your function or form should not be very large. That means then anything can happen, right? You don't want promiscuity. You want there should be specific things, right? You think about receptor ligand interaction. You don't want my receptor should interact with hundreds of different receptors, ligand, right? You don't want promiscuity. You allow some amount of promiscuity, but it's a narrow range, right? Similarly, if you are creating any model, cell signaling model or anything, if you see you have a very large parameter space which is allowing you to do it, then you are actually not really in a biological grid, because it is eventually makes promiscuous. At the same time, you remember, you cannot make the space very narrow. Then there is no scope of evolution actually, right? System cannot evolve, even it cannot adapt also. Fine. So that means there must be a balance between this opposing requirement in terms of parameter in a real biological system. In one end, I want it to be very flexible in the changing environment, but in the other end, I do not want it to be so flexible that any damn thing can happen, right? So this data is unsettling. That means this is not actually real one. Yeah.
Can you get alignment in the right direction even when M3 is 0?
No, this is a very narrow band here, which is not blue.
So what happens when M3 is 0? What's the color at the lower right?
Yellow to green.
Shouldn't it remain yellow in the sense you shouldn't get any alignment at
No,
all?
because you have from 0 on the line of 0, it is yellow.
Right, okay.
But then you have small values
Okay,
which
okay.
get compressed, right? So the scaling issue is there. I cannot capture the whole thing because we have very, the deltas are very small. So this is important. So then that means, although in my model it is happening, but in reality possibly this whole wide space is not available to the system. Even if the system, I consider that it is working like that, right? So then the question comes that this is the first thing. Second thing, as I was saying, that you can see here again, if M is 0, M3 can achieve complete alignment, job done. So as I said, this is one unsettling question in every field of collective behavior, that if a global Q can do it, you have a diffusive molecule across the system, and that achieved the whole alignment. Then why you require local, right? Why do I require local? There are many ideas. For example, in case of organismal behavior, people say that actually if you want that, you should have a global authority which is permanently authority. So from the developmental time, you have to switch on something and you have to create the authority. Whereas if you allow only local thing to interact, then you are not creating any other separate organism. So in terms of cost of maybe this is much easier. I do not know exactly what the reason here, but these two things triggered us that to look into what may be the role of global thing when the local can also do it, whereas the global can alone also do it, right? Let us look into this. For that, we moved into a system where we mutate some of the cells. What do I mean by mutate? Here, I have the lattice where cells are wild type, some of them randomly placed anywhere. Those mutated cells, they cannot sense the global Q, means they cannot sense the global field, right? So I am mutated, you people are wild type, you are my neighbor. I do not know which way I have to align, right? I have interaction with you, so interaction with you should eventually help me to get aligned in the right direction. That's the spirit. So the question is, see in this plot, this plot actually is misleading because see, in terms of biological function, you need everybody to get aligned, right? So what we do, we make that read of the plot. So this black region is where Fu is bigger equal to 0.99. That means almost all of them are aligned. That's what we want, right? So this is wild type, and now you can see here you have this region, right, where you do not have the alignment, which is close to 0, which is much more clear. Now what do we do? We mutate 5% of cell. Then we mutate 10% of cell. You increase the mutant, but you still see a parameter region where all cells will get happily aligned, including the mutated one. So that means what is happening, although those cells, 10% cells, they cannot sense the global Q, still they get aligned because of the pressure of the neighbors, how the neighbors are telling them to get aligned, right? But that makes sense, but look into how this is shrinking. This part is getting shrink, isn't it? Not like here something is getting shrink somewhere. Why it should happen like that? Let us look into the previous model data. In the previous model, I have a threshold, right, which we call percolation threshold, PC, right? So similarly, I have a FC, M value, which is the critical value. So this is the critical value of my earlier model, where I do not have the global thing. So in that case, when you cross this value of M, or you are near that value of M, you have a global order already formed because of the local interaction only, right? So there is a long distance correlation. When you forget about M, when you have a long distance correlation when you are here or beyond that. But that can be in any 6 direction. Now, if you are above this threshold and you slightly give a nudge by a global Q, then everything is favored in that direction. Whether I am mutated or not doesn't depend, because there is a global order. We could have all aligned in any possible sticks direction together, mutated and wild type, but you have given a nudge by the global Q and we all align in the right direction, and that's why the shrinkage is happening here in this region where you do not have global order by itself by the local Q only, right? And that's what it happens. And in fact, this is very interesting because we initially showed how this behave in another numerical way, how much is the deviation from the wild type behavior. So 5%, 10%, you can see 2.223 is actually the threshold. Before that you have quite difference between 5% with the wild type, 10% with wild type, but then the wild type and those error is almost zero, deviation is zero. Now the interesting thing is that our reviewer said that, maybe passing remark, what happened when I have extreme mutation? Extreme mutation mean what? Even 10% mutation people will not try in experiment actually. We said, okay, we have a thing, so we can have even 95% mutation, and you can still see the bottleneck is still before that. Once you are beyond that threshold, actually the global order is so strong that even majority of them are mutated, still they will get aligned in the right direction, right? So far so good. Then interestingly, another reviewer suggested why not gradient, why you are considering a uniform field. Yes, that's a quite logical thing. What will happen if you have gradient? So now we consider a gradient. These
I have a question, sorry.
Yeah.
So in the case of wild type, once you give a nudge with the external field, I suppose you can switch it off later because the system has already started aligning.
Right. What happened to that?
Right?
I'll come.
But I suppose the result wouldn't be different.
It can.
In wild type, meaning
wild type.
without mutant?
Yeah, without mutant also.
So you begin with a field
If
on
there
and
are,
then...
there are two ideas in this field. What you are saying, I'm coming from a biology perspective, I will say that initially there is a global Q and then it disappears. Right? That's what you are saying, right? Huh?
Without considering biology for now, just sticking to the model.
Yeah, I'm sticking to the model. What will happen in this case I will show, but there are a model where if you remove this global Q after getting the equilibrium, right, or the steady state breaks.
But you can have global order even in the absence of external field, right?
Yes, but then it gets aligned in another direction.
No, but once it has been aligned by the presence of field initially, so you start off with a field on,
Yes.
you keep on increasing the parameter that takes you to the, which causes phase transition. So the parameter here being m.
Yes,
Right?
yes.
You start from disordered, the field is on, you cross the critical point, the field is still on, then you switch it off. The system should still be aligned. I mean,
Which one, this M or M3 you are saying?
for the case of wild type, where you don't have
which one you
mutants.
want to switch off, M or M3?
M3. Is M3 the external field? I don't know.
M3 is external field.
Right, you switch off M3.
In our case, it will stay what you are saying. What I am saying, I will show that it stays, right?
Right.
But there are PDE based model where if you remove the external Q, that doesn't.
You're right. So no, I asked this because I was wondering if you will see different.
I'll give you the answer beforehand. This is our data. So what we do here, we start with a M3 value, particular M3 value, right? We simulate, we know up to where the equilibration starts, right? So we reach up to that point and then we draw M3, make it 0, right? So that means what you are arguing, that I am already in equilibrium, and if I remove the field, do I still stick to that same equilibrium or not? That's what you are asking, right? And yes, in this model that happens.
Right. So I asked this because I was wondering if you would see the difference in the presence of mutants.
Okay, that I have not tried, but it's a good idea. But actually this is very interesting in a sense, I thought I will tell you later on. See, this is actually possibly what is happening in biology also. See, remember the whole dispute with global Q is maybe problem of detectability. If something is happening very early and very weak, my experience with experimental biology says I will possibly miss it, right? Even if it happens in, suppose in this scale, maybe our scale, but imagine you are doing cell signaling experiment where the phosphorylation happen within 5 minutes and then everything disappears. It's very difficult to, where I will do 5-minute sample, 10-minute sample, I will get wrong, right? So possibly in biology also, maybe not in second minute scale, in this case hour scale, but we do not know beforehand, right, where to pick the sample, right? So that may be happening actually, the time I am considering for my experiment is already the system is there. the global Q was very early and it has pushed it to the equilibrium and then has left. That may be the reason, that's why biologists are missing it also. This is one possible reason. Fine, clear? That does make sense? Okay. So if I go back to my thing, so this is, that's a good point. I will just think about how we can put, right? So now, what we see here is that,
Sorry, can I interrupt?
yeah, yeah.
Also, in the slide where you showed that you vary the mutation fraction over a wide range, so why do you see some peaks for?
Yeah, that I don't know. That's why I put this one here. If you can, what you can suggest.
I see.
I am not revising the article because I am stuck with this. I do not know why the peak is coming. Initially, I thought I may have done a mistake somewhere in the calculation, but no, it is not. There is a peak.
I see. Okay, thanks.
Right? So in our archive paper, you do not have. In the revise, I have to put it, but I am still stuck. I don't know how to do it. Sorry?
Do you think it might be a finite size effect, that peak?
Finite size effect. How come?
As in, if you scale the system size, it might go away.
Scale in the system size in the sense?
If you keep increasing the system size, you'll see that the bump, that peak, either shifts to zero or sort of
Okay, that
goes.
will be the way to test it. That's fine. But what is the rationality? Why should I test that? I mean, I can do 500 lattice size. But what should be the first point? Why it should? I mean, any clue? I mean, I do not. If I have a clue, then I can try, right? And yes, if you NH size effect is the simplest way, you run a simulation, you check it whether it is affecting or not. But what should be rational? I do not know. I mean, okay, we'll discuss.
Yeah.
This is a sticking point actually, I'm stuck, right? And I am very lazy on work, so it is stuck for a long time. And I hate to rewrite papers actually. So if you see here, we have a gradient. Alpha is the parameter, so I can make alpha 0. That means in this row there is no M3. So this one, and in that case also you see the similar behavior happening. If you are beyond that threshold of local calculation, you are actually safe, right? So let me jot down where we are going. This one I have already discussed. I will skip those things. So let us come back and let me jot down, right? Almost there. See what is happening then. If I look into my system, with the second model, we can easily see that in this case, you face a situation where you go ask why you require this global one. Fine. And then if the global one can happen, then why you require the local one? The easiest answer to this is that, see, the local interaction. If you forget about any other cost, how to make all these things, local interaction allows you, right, to make the system robust in the phase where the global thing will fail, definitely, because the global one does not give you a long range correlation in that sense, right? And if you think in terms of engineering principle, the chance of failure in the global one can be very high, because the global one may be getting secreted or created somewhere else, right? Some global authority, right? So although the global thing, you can achieve this alignment in the global one, but you can actually create an evolutionary system which may have evolved where each cell has the same machinery to do the local interaction, and so you are fail safe because it is genetically encoded. So you are just repeating it, right? And this local interaction actually can achieve alignment. Only thing what you require a very small nudge, very small nudge. You can see here, if you give a very high local interaction, then slightly above 0 global signal can actually achieve it. And suppose I am starting the signal from here, and this whole thing is my lattice of cells, it may not have to go up to that end. Even if it goes slightly up to this, as we are saying for mutant, right? Because of this local interaction and the strength is high, everything will get aligned. So I am quite fail safe. And at the same time, as I was saying, arguing for those data where actually we can say that actually you do not need to give this type of system, keep them global Q continuously on, you can switch it off also, right? Once you have reached that thing. There are ifs and buts. I cannot vouch whether it will happen in a real system or not. You can always argue the real system may not be such equilibrium system, not so simple, whatever it is. For this model at least, we can argue that in this case at least we have, and remember that those PDE based model actually fails to show that. They require constant external Q actually, global Q. But this tells us both these two things, that actually with a very weak global Q, as long as you are above this percolation threshold, you can achieve the global alignment in the right direction with a global weak Q and a transient global Q, right? And that can be weak even in case of transient. Now these two are perfect recipe in your experiment to fail. As I was arguing, your global Q is short term, you miss time, you cannot detect. Even if your time is perfect, global Q level is so weak that your conventional experiment fails to detect, right? So these two are possibly the reason that biologists are struggling to identify and fighting on the issues that what should be the global Q. The nudge may be very slight and maybe for a small time. The key issue is this local interaction, which is very predominant, and it must be above that percolation threshold. That's why every time you do an experiment with mutation of those, your experiment succeed, and you so early detect is this local module, right? That's the takeaway for this thing. We have tried making non-equilibrium, breaking the Dittel balance. You really do not get some interesting thing, otherwise I would have discussed that, right? So that's what we learned. I will just want to end with one thing, that remember this is the behavior of our model. Don't map it one-to-one with what you see in the Drosophila wing, right? Drosophila really doesn't care about your percolation. That's all. Thank you.
Yeah, so any questions?
What would happen if M2 was set to 0? M2 is the interaction within a cell, right? The coefficient.
M2 you require. M2 require within the cell.
So just a strong M1 won't.
No, M2 is required, and that is possibly, that's why actually you have, we have tried in other way also rather than considering external fields. See, actually inside the cell you will see movement, diffusion, facilitated diffusion of these molecules happen, right? And those are also controlled by cell-to-cell signaling, all these things. So if you switch up those, if you disturb those experimentally also, then it is dead. Everything goes wrong. In our case also that. M2 you cannot. You can make M1 cell-to-cell interaction off, but keep a very strong inside segregation, then you will get it.
Also, the cell-to-cell communication pathway that you showed, is that something specific to Drosophila, or is it like common across many?
The Frizzle band pathway is conserved across up to us, right? But it may not be working in exactly this fashion in different system, even inside Drosophila in different cellular system, but these molecules are required.
Okay.
And we have homologs like Frizzle. I think there are 4 Frizzle in case of Drosophila, we have 10 of them. We do not know most of the functions and all this thing. There are case of interesting case also, some of these molecules have multiple homologs. They can do the same function, but why they are in the Drosophila, we do not know. Okay. But very conserved.
So can you repeat what you argued at the end, that about the weak morphogen and global?
So what we are showing, if you look into the state space thing, the key is that your M value, that local interaction thing within the cell with the neighbor, that is a local, these two make local, that if you can keep above the threshold, their percolation threshold, then even a very weak M3 global field can allow you to get aligned in the proper direction, right? Second argument is that from that phase diagram where we have done simulation, where we keep the M3 global field only transiently. Once you push the system in equilibrium, what we call after the burning phase, you withdraw it. So it is once in that equilibrium, it stays there, right? So what does this mean? That means that a transient weak global Q is good enough for the whole system to align in the right direction and polarize collectively, given your local interaction is strong enough and beyond that percolation threshold. That's what I was saying. And the argument is, if you have this type of situation, then it is difficult possibly and experimentally to pinpoint what is the global Q.
So can we say that this is biologically relevant also, because in stages of say wing development there could be a situation that the Q would disappear eventually, but before that,
that's the whole idea, right? That's the whole idea. The thing is that how it will disappear is the issue. For example, for in this model also, if you think like that, you will create with a one layer of aligned cell, like this wing is growing, right? So all the wing is not simultaneously there. So you imagine that initially this layer is aligned, and then I remove all global Q. Will it, this one seed will help this to align? No, that doesn't work. So the issue is how this global Q works and how long it stays. That's also important, right? So definitely it is true what you are saying developmentally. Somewhere the switch happens, some events happen, and then possibly disappear. Just like we have the cell cycle commitment, we have the bifurcation where we have decided that we will do, you cannot go back, right? Even if the growth factor you withdraw, you have to complete the cell cycle. Something similar may be also there. And that makes sense in terms of the struggle that people are doing to detect them. That makes sense.
We have time, so.
Just one quick question. I have a couple of questions, but I'll ask you later. Just one quick question. So, the exponents that you got for the finite size scaling, so you got that from just 3 data points, I suppose, because you have just 3 sizes, system sizes.
Yeah.
You think if you do it for more sizes, the exponent values will improve? Meaning maybe even get closer to the...
Actually, these are good enough, if you ask me.
No, I agree.
Look in the error term, right?
Right.
And it's not a negative situation in that sense, right? And what I was more happier is that, see, when we take the leap of faith that you don't have a P, right? We are just imagining that then there must be these two things are equivalent with a constant somewhere, right? That's you are taking. You don't know. I don't have a proof, right? I'm just believing that it must be scaling with that, and it worked. I'm happy, actually, right?
Okay, so any more questions? Okay, if not, let's thank the speaker once again. There should be tea outside, so we'll break for tea, and we will return back here by 12 for the next session. Thank you.
So welcome back, and today, we have this 2 talks on the biology of dictyostelium or slime mold. You remember that we had this modeling talk yesterday, the last talk of yesterday, where Trilochan showed, a model of how dicty does cyclic AMP signaling, and there's a streaming motion, and then this bud. This slug forms and so on. So, but he didn't go into the biology at all, and that's because Bhaskar and later on Professor Nanjindia is going to give details of this. So it's a pleasure to introduce Professor R. Bhaskar from the Department of Biotechnology, IIT Madras, which is basically our neighboring institution. So Bhaskar did his PhD with Professor Vidyanand Nanjindia at the University of Science, Bangalore, and then he went for his postdoc at Cold Spring Harbor Lab, where he worked with Professor Uli Gross-Nicholas, who's also visited IMSC actually a couple of times, right? Thanks to Bhaskar. And then after a short stint at NCBS, Bhaskar joined IIT Madras in 2004, where he runs this plant lab, which looks at very exciting things. And it's been a pleasure to, discuss several times with Bhaskar on his very interesting projects. And today, Bhaskar is going to give a talk which is partly going to be introduction to the dictyostelium biology and partly going to talk about his own research. So without further ado, let me invite Professor Bhaskar for his talk.
Thank you. Thank you so much, Tabra. People in the last row, can you hear me, please? Yeah, good. So we work with 2 fantastic model organisms. One is dictyostelium. This is a social amoeba, and also a model plant called Arabidopsis. And polyspondylium is the model that I started but later gave up because of various constraints, because this was not easy for us to transform. And I'm afraid that this talk is going to be completely out and out biology. If you don't understand at any point of time, please don't hesitate to stop me. So I'm sure you must know, some of you may not know, or some of you may know what is embryonic organizers. This is a small subset of region within a developing embryo, a vertebrate embryo. The red region is highlighted here. If you transplant this portion of this, the red portion, and then transplant it to a chick embryo, another embryo, so finally you get 2 head formation, finally 2 head formation, which is there in a different model here, chick model, as well as a model, it's an amphibian model. So this region, the red region that is highlighted here, this is called an organizer. The organizer is a region of a subset of cells within a developing embryo, vertebrate embryo, which induces the neighboring cells to take on a different cell fate, and it gives you a polarity, it establishes the polarity. Without an organizer, the organism will not be developing further. So if you have a block in organizer development, completely the development will be blocked. And the second term that I want to introduce is about morphogens. This morning I heard someone talking about morphogens. I'm sure you must be knowing what is a morphogen. So these are gradients of substances along the length of the embryo, which is established and stays as it is. So now you can ask me a simple question: can the gradient be, because of diffusion, can this be diluted out? But that doesn't happen. There are various mechanisms to restrict this gradient, and the concentration gradient of these embryos, concentration gradient of these morphogens along the axis of the embryo, developing embryo, determines the cell fate. For example, the cell fate, that is the fate X, Y, and Z, that depends on the concentration gradient of these morphogens along the axis of the embryo. So now comes dictyostelium. I want to run a short movie here. Some of you may have seen this. This is a motile stage. This is a multicellular stage of dictyostelium, which is mobile on a agar substratum. This is called the slug. This movie is from John Bonner, who was a pioneer in dictyostelium research. This is a single-celled amoeba, which is grabbing its food and then looking for food. So now they are running out of food. If they run out of food, something interesting happens. All the amoeba converge at common collecting points, and then because of cyclic AMP chemotaxis, because of signal relay, one amoeba attracts each other and then forms a structure called an aggregate. So this aggregate of cells slowly turns into a motile structure called the slug. So now the aggregate becomes what is called as a mound. The mound, then it undergoes further morphogenesis, and then it becomes what is called as a fruiting body. So the 20% of the cells in the slug, the anterior 20% of the cells, roughly the size, roughly the anterior 20% of the cells, they go through programmed cell death and forms a dead stalk, while the rest 80% are lifted in the air and forms the spore mass. So this is another movie, which is a very nice movie I want to show. So here you add this is the bacterial plate, here you add a drop of dictyostelium cells. So they eat and forms a fruiting body. Those are the next round of cells, it forms like this. This is very cinematographic or whatever. So you have a colony of dictyostelium. If you initially spread a lawn of bacteria, eventually all the lawn of bacteria will be eaten away, and then these slime molds, forest of slime molds will be formed. You get fruiting bodies here. Here you have, so what you have is a circular plate. In the center you drop a piece of dictyostelium cells. What happens is they keep eating, you know. If they keep eating, then the circumference also goes up, no?
Yeah, so that's that visible wavefront that we are seeing, is it like the depletion of the bacteria?
It is absolutely depletion of bacteria.
Oh.
So to summarize the life cycle, so as long as there is food source, they keep eating and proliferating. So once if the food source runs out, something interesting happens. So the amoeba becomes aggregation competent. So the cells converge at common collecting points. It forms what is called as a mound. This mound slowly forms a structure called as a tip mound. The tip here is functionally equivalent to the embryonic organizers of higher vertebrates. Then the tipped aggregate slowly turns into a structure called a slug. The anterior 20% of the slug, they die and forms a dead stalk here, and the rest 80%, these are called pre-spore cells, they form dormant spore mass, spores. So the embryonic organizers, the functionally equivalent organizers of higher vertebrates was discovered in the year 1975, and if you transplant the tip of dictyostelium slug to another slug, you eventually get two-headed slugs, two slugs from the single body. So at various sites this transplantation was done, and they found in all cases there are two slugs emerging from a single slug. So this was identified, even though organizers were largely confined or originally discovered in vertebrates, this was also functionally equivalent organizers were also identified in dictyostelium, and the various morphogens along the slug axis were the well-known morphogens include cyclic AMP, adenosine, ammonia, and DEF. Cyclic AMP is a chemoattractant. It is also a first messenger. Everyone knows it's a second messenger, but here it is acting as a first messenger. Then the breakdown product of cyclic AMP, the cyclic AMP is degraded to adenosine, but there's little bit of controversy whether adenosine is a morphogen or not. Then volatile ammonia, ammonia is a gas, and volatile ammonia is also acting as a morphogen, and differentiation inducing factor, it's a chlorinated hexaphenone, that is also acting as a morphogen. Cyclic AMP and ammonia, they are known to promote the pre-spore forming pathway. Adenosine, it's still controversial, whereas DIF, differentiation inducing factor, it promotes the stalk cell forming pathway, the stalk here. So what the structure that you're seeing here is approximately 1 millimeter or anywhere between 1 millimeter to 4 millimeters or so. So my research right now, what I'm going to talk to you today, where is this ammonium coming from? The role of ammonia has long been recognized in dictyostelium. So this is a picture that I took from the paper published several decades ago. So there's a gradient of ammonia that was known to exist, the gradient of ammonia, whatever. So where is the source of ammonia for all this dictyostelium? Because ammonia is also toxic in certain, but ammonia is produced in millimolar quantities in dictyostelium. And where is this ammonia coming from? So when they quantified proteins, overall level of proteins over a period of time, the proteins levels were dropping over a period of time beyond a period of starvation. So subsequent to starvation, if you quantify the total proteins, the protein levels were coming down, and then they assumed that the degradation of proteins is responsible for the origin of ammonia. And this was for a very long time, this was the view, and now we have a different view also. Maybe both of us are correct, we do not know. So adenosine, adenosine is derived from cyclic AMP. Adenosine is derived from cyclic AMP, and there is an enzyme called ADGF, adenosine deaminase related growth factor. Adenosine deaminases are enzymes which acts on adenosine, releases ammonia as a byproduct. So this ammonia is responsible for the further morphogenesis, or is acting as a morphogen. This adenosine, this is called adenosine deaminase. This is adenosine deaminase related growth factor. This adenosine deaminase related growth factor is extracellular. It is made outside the cells. So it is, the enzyme is thrown out of the cell, and it is acting on the extracellular adenosine. The ammonia is formed extracellularly and drives tip formation or drives organizer formation. Then we wanted to find out, so when we started this project, there are many deaminases. Deaminases means which acts on the amino group and then releases ammonia. So which deaminase to work on? So one of the deaminase that caught our attention was adenosine deaminase related growth factor, just because of the fact it is extracellular. And in dictyostelium genome, there are 4 adenosine deaminases annotated, and this is the adenosine deaminase that we are working on. There are 2 independent insertions here. This is a blastocidin resistance cassette which is integrated here. I'm sorry. There is one insertion, there are 3 independent insertions here in this adenosine related growth factor gene, and all of them give the same phenotype. So then we went on to, then we pursued with this mutant. We obtained this mutant from the stock center. It was not created by us. So now we'll run through another movie here. Do you see this? This is aggregates. First of all, what you see here, this is a mutant. The mutant is slightly larger than the wild type. This is the wild type, and this becomes a lox. So it spins on its own axis like Vishnu Chakra, and it keeps on spinning and then spinning and spinning for a very long time. Then it gets arrested. And a fraction of them go on to form fruiting bodies after 36 hours or after a very long time, a fraction of them goes on to form the fruiting body. So I keep on emphasizing this. So what is happening here in this mutant is you have a block here. If you have a block here in this pathway, then you expect adenosine levels to go up, ammonia levels to drop. So is it because of high adenosine levels this is happening, or is it because of the absence of ammonia this is happening? So now these are series of pictures. What you see on the top panel here, this is the developmental profile. This is the wild type. This is called the mound. It is going to form at 16th hour. It is already showing signs of fruiting body formation. So by 24 hours it has formed fruiting bodies. That is a terminally differentiated state. Whereas this is the mutant. The mutant is there as a mound, continues to stay as a mound for a very long time. So they are not dying. So a fraction of them, they form fruiting bodies after a long period of time. You can mimic this phenotype by having a ADA inhibitor. This is the inhibitor which is very specific for the ADA enzyme. If you add this inhibitor, then you essentially mimic the same phenotype. So that is the first line of evidence to show that the gene that we are working on is indeed the correct one. Then the second line of evidence is this one. So this is the developmental profile. Is it not moving? Yeah. So we have to confirm that the mutant that we are working on is not showing any expression. That is a confirmatory test. That is, we are working on the right gene. The gene that is in the wild type, it is the peak in expression comes at 16th hour. It is roughly corresponds to the tip formation, the organizer formation that happens at this stage. And we wanted to see whether the other isoforms of ADA, These are 3 other isoforms of adenosine deaminases. Are they trying to compensate the loss of this mutant? Are they trying to, because in the absence of ADGF, these adenosine deaminases can try to compensate the loss by increased expression. So when we checked it, it's not happening either. By RT-PCR we checked it, it's not happening. Then coming back to this, so what is happening here? Here you have a block. If you have a block, this block in this, adenosine levels are going to go up and ammonia levels are going to go down. So we checked the adenosine levels using a commercial kit, and the adenosine levels at 16th hour, it's significantly higher in the mutant compared to wild type. This is total adenosine, this is not extracellular adenosine, this is total adenosine. And the ADA activity also comes down. The ADA is the adenosine deaminase. This is the enzymatic activity, it drops drastically. It is not completely abolished because there are 3 other isoforms which are still have a basal activity here. That may be the reason why you still have a basal activity. Do you see a movie here? Do you see a kind of, once again, this is a cyclic AMP. So cyclic AMP is one of the morphogens identified in dictyostelium that plays fundamental roles till tip formation, till fruiting body formation. So we wanted to check whether what is happening to the cyclic AMP levels here. The cyclic AMP levels are low here in the mutant compared to wild type. We measured at different time points, and 2 different time points at least the cyclic AMP levels drop drastically. Here we can visualize the cyclic AMP wave pattern also by dark field optics. It's an indirect way to visualize cyclic AMP. For example, if you visualize under dark field optics, what is happening is cells which are elongated and cells which are round, they are going to scatter the light in different ways. So one you are going to see the light bands and the dark bands. So based on this, over a period of time, if you take a movie, then you will be able to see such man. Here you can see a spiral wave, whereas in the mutant it is a circular wave throughout. So initially they start as a circular wave, then there is a symmetry breaking happens, then it forms a spiral wave. That doesn't happen in the mutant at all. So ADGF mutants also forms large size aggregates. Why dictyostelium also people use this as a model organism to know how tissue size is regulated. An aggregate or a mound or a slug is of certain size. If there are genes going impaired in this pathway, the size regulation is something goes wrong in that process, then the size becomes bigger. So ADGF mutants form large size aggregates. As you can see, the size, when you have a large size aggregates, obviously the number of aggregates comes down, and it is because of, we found this was not the main focus of our work. We found what is the reason for increased aggregate size. It is because of increased cell addition, and there are genes impaired in involved in genes involved in tissue size regulation. Their expression, these are some of the genes involved in tissue size regulation. Their expression is altered, as a consequence of which you have the size goes wrong, and then you have a large sized aggregate. Then coming back to the mutant, what is happening here? What is, we wanted to check further what is happening to the relay, cyclic AMP relay. There are various genes involved in cyclic AMP relay. So the first cell that undergoes starvation makes cyclic AMP. The second cell perceives it. Then it relays the cyclic AMP signal to the third amoeba. Thus all the amoeba come together. So there are various genes involved in this pathway, various proteins involved in this pathway. So we looked at the expression of various genes. This is called a phosphodiesterase. This is also a phosphodiesterase. This is also a phosphodiesterase. These 3 phosphodiesterases, one of them is membrane bound, one of them is intracellular, one of them is extracellular. These are phosphodiesterase. This is 5' nucleotidase, which is involved in adenosine formation. That expression level, so this generates this. If this gene is very highly active, you get excess adenosine, and that is what is happening here. CAR-A is involved in cyclic AMP receptor. GBFA is a transcription factor involved in tip formation. So we looked at various genes, their expression profile over different time points. And coming back to the wild type. So now is the wild type, that's a mutant which you can see. Now can we take the supernatant. The supernatant, we call it as conditioned media. Can we take the supernatant of this wild type and see if we can add the supernatant to the supernatant, the AX4, for example, this is AX4 supernatant on AX4, this is the control. The same control goes here. Whereas if you take the wild type supernatant and then add it on to the mutant, then it gets rescued. Similarly, if you take the conditioned media or the supernatant of the mutant and add it on to the wild type, you have the same developmental block. So something is secreted, and if something is secreted, as a consequence of which you have this problem. If something is secreted and if the secretion is not proper, then you'll have the problem like this. You can also mix wild type. Here it's a medium supernatant. You can also mix the wild type with the mutant, physically mix them. And if you mix them in a 20 to 80% ratio, you have partial rescue, whereas 50 to 50% you have a complete rescue. Now, is it because of adenosine deaminase? So we took the mutant here. Here is a mutant directly on top of these mound arrest. These on top of this structure. We add the human ADA enzyme. This is a commercially procured enzyme. So this enzyme directly we add it onto the top. Then it is a rescue. So if you have more, you add it. It's complete rescue. This is also getting rescued with caffeine. What does caffeine do? Caffeine is an antagonist of adenosine. Antagonist of it, that is also getting rescued, and this is 8-bromocyclic AMP. 8-bromocyclic AMP is a stable form of cyclic AMP which gets into the cell and stays for a longer period of time. That is also getting rescued. So this is already suggestive that cyclic AMP is the main player here, and cyclic di-GMP also rescued. The reason why I emphasize cyclic di-GMP is because of cyclic di-GMP is a prokaryotic signaling molecule. And that is also used by eukaryotes, and that induces the organizer formation, that induces the pre-stalk formation. That was published by someone else many years ago. See, when we checked with cyclic di-GMP, that also induces, but as the concentration goes up, you can see some multiple tips. So instead of one tip, you get multiple tips also. Now, if you go back to this reaction, adenosine, it forms ammonia. So if you have a block in this pathway, then ammonia is not there. So now we ask the simple question, is ammonia, can it rescue the phenotype? So what we did was, here you have a plate like this where you have the mutant. On the top lid where you can have here you can generate ammonia artificially. If you generate ammonia artificially, then what happens? There is a rescue which is happening. So in all the concentrations we tested, the ammonia is getting rescued, and the ammonia levels, when we tested, the ammonia levels are also lower in the mutant, but when we add it externally, so the ammonia is not physically, it is there in another lid, so found it is able to rescue the phenotype. Now this is an interesting aspect. Now wild type without physically touching the mutant. So wild type, what is happening? Wild type should produce ammonia, whereas the mutant is not producing ammonia. So what we did was, we took a compartmentalized petri dish. On one side we have the mutant, on the other side we have the AX4, which is the wild type. AX4 is the wild type. Now we asked a simple question: if it is genuine, if ammonia is indeed responsible for the rescue, can it rescue the phenotype here? Because here you have a compartmentalized dish, so it is able to rescue the phenotype here. So without physically touching. So this is happening. And what is interesting here, now we asked another question. I'm not sure if I have the slide, but I'll come back to this later. We also added bacteria here in the one side of the petri dish, on the other side the mutant. Bacteria, if you have bacteria on a non-nutrient agar, the bacteria is going to die. When they die, it is going to generate ammonia. The dead cells generate ammonia. When they generate ammonia, it is going to rescue the phenotypes. So bacteria also rescues, but after a certain time. Wild type rescues in a period of 4 to 5 hours. And this is the idea of conditioned media. So if you can take the conditioned media, that is the supernatant, you can add this ADA enzyme and see, ask the simple question whether it rescues. There also it is rescued. Finally, the gene, the overexpression, if you take the mutant and then compensate this with the overexpression construct, you take the wild type copy of the ADGF and then put it here, then that is also able to rescue the phenotype. So multiple lines of, yeah, please.
Can you once again explain the setup in there, in that sense? Like
Yeah, this is a partition petri dish. That means there is a physical compartment running through. That is what you see here. On the one side you have the mutant. On the right side, on the left, for me it's right side. Yeah, on the right side it's a mutant, left side it is a wild type. Now we ask a question, is if ammonia is produced by the wild type, can it rescue the ADGF mutant? And the rescue happens.
So ammonia is diffusing through the partition?
No, it's not completely sealed.
Okay.
It's like the porous structure here, what you see on the top. The ammonia goes low.
So apart from ammonia, other morphogens can also diffuse?
No, through the air.
Through this partition. Oh, okay. Ammonia is volatile, yeah.
Ammonia is volatile. That's
okay.
why it's able to rescue. The others are not.
Okay, thank you.
Yeah. And I'm not, the temperature is at 22 degrees Celsius, so it should not happen. Yeah.
By looking at the images, is it easy to see if the mutant has been rescued?
Yes, it is rescued because I see the tips here, several tips here.
I see.
And also the size slightly goes down, you know, because the size is here, it's all, you can see these are all like pebble stones, no? The pebble stone, you no longer see that kind of pebble stones here. Now, we have not proved, so far we have not proved, so even though I took the wild type and then have the mutant on the side, we have not proved so far that adenosine deamination, that's a chemical adenosine, from the adenosine there's the amino group is cleaved off and then it is responsible for ammonia formation. So similar to the last experiment, here we took the mutant, on the left side we have the buffer. So the buffer, it's the ice cold buffer, to this we add the ADA enzyme. ADA enzyme plus adenosine. So on one side you have the adenosine plus buffer, and the right hand side you have the moment you add the buffer, after 4 hours you get, this is getting rescued. So all of them are rescued now after a few hours. So this convincingly demonstrates that adenosine deamination alone drives the rescue. So downstream of it is cyclic AMP. So now we took several mutants which is having this kind of phenotype, like pebblestone kind of phenotype, and then took these mutants because they show a very similar to, why do we take this mutant? To know what is the pathway by which this gene works, the ADGF works, pathway, by which mound formation, tip mound formation or organizer formation takes place. So we took several mutants of this, mutants which looks like this, and then directly added ADA enzyme on top of it. When we added, then we found that this is a histidine kinase. Histidine kinase is, this is a mutant impaired in histidine kinase. If you have a block in this, then it is manifesting in mound arrest. So if you treat them with the ADA enzyme, then it is, you are able to see a rescue. The more you add it, the more then you can see a multiple tip phenotype. So this is a pathway by which we are able to find out. We tested several mutants, of which only we found DHKD, and these are other mutants. When we added this ADA enzyme, there was no rescue. Similarly, what we did was, we took mutants with multiple tip phenotype and added, then we did the reverse. We tried to block the ADA activity by adding the chemical inhibitor. When we did this, there is no rescue either. So it's of course, we have not comprehensively looked at all the mutants available. We took a range of whatever that is, half a dozen mutants or so. Now, DCF, so is it affecting the pre-stalk, pre-spore pattern here? The pattern, that what, already this is the pattern, the pre-stalk, pre-spore pattern is, what is happening here? So this is the slug. The front end of the slug is going to die and form the stalk. Here you have a molecular marker. This marker is called the extracellular matrix, and that is tagged to GFP. And when we had the drug, when we chemically block this ADA with a drug called DCF, then you see a slightly enhanced expression, and you also see a slightly some pattern of expression at the back of the slug. Conversely, you can take the pre-spore, that is the other end of the, so this is pre-stalk, this is pre-spore in the wild types. So what I see, this is the front end and this is the back. What you see here is the front is the pre-stalk. What you see on the back is the pre-spore. So now when we did this for pre-spore, what is happening here? This is the wild type. After adding the drug, there is no change in the pattern. But when you take the neutral red marker, this is the front side, this is the front side, and this is the back side. But in all of them, there is a slight, in the fraction of the slugs, there is a small bit of increased expression, expression of pre-stalk cells on the back of the slug. And you are able to see when the pre-stalk marker expression, the ECMA marker, which you see here, that is the spatial pattern. There is no change. But however, there is a slight increase. There is a reasonably increase in pre-stalk expression in both the cases. Probably these cells may account for the increased expression here. And pre-spore expression is down-regulated in both cases. Now, ADGF cells have high levels of adenosine. The view is that high levels of adenosine at the slug front is suppressing one more tip along the axis. So we took cells with high levels of adenosine, that is the mutant. This is the wild type on the top panel. We are labeling with a cell tracker dye. This dye completely stains the slug with red color, which you can see here. So this is the wild type. This is the mutant forming a large colored slug, large size slug. When we the ADGF cells, which is carrying high levels of adenosine, when they are mixed with the 80% of the wild type cells, the cells carrying high levels of adenosine goes to the slug front. Conversely, when you do the reverse experiment, you can see scattered staining here. Now you can ask me why the staining is scattered here. Because we are doing with 20% cells here, not 80%. If you add 80% labeled cells, then the slug will be glowing. Then it will be difficult to visualize the fluorescence. Now, how is this gene regulated? The gene has to be regulated by some means or the other. So we took the wild type cells, this is wild type cells. They are treated independently with the adenosine or ammonia. This is the substrate, this is the product. Then we looked for the expression of various genes involved in this, various genes that is of interest. One is the adenosine deaminase, the same gene that we are working on. This is in wild type. Then this is the gene that is responsible for cyclic AMP synthesis. And this is the gene that is responsible for cyclic AMP degradation. What is interesting here, which if you can draw your attention now, here is you see that from AX4, as the concentration of adenosine goes up, there is a gradual decrease in phosphodiesterase expression with the increasing levels of adenosine. Whereas with the increasing levels of ammonia, the levels go up, completely contrasting expression. And ACA expression, which is responsible for cyclic AMP synthesis, depending on the dose that you can see, as well as the ADGF, depending on the dose that you see, there is differential pattern of expression. So now we asked how if adenosine is involved in suppressing tip development, if you add adenosine to wild type, will it suppress tip development? Adenosine is responsible for, that is the hypothesis we start. However, when we added adenosine, it is not doing substantial changes to the tip formation, no change here. Adenosine after 10 hours, no change in the wild type. The derivative after the, if ADGF really works, inosine is also formed. Inosine also has no effect. Related purines we also checked with guanosine and deoxyadenosine. They also don't have any, they also don't exert any effect at all. So mutations in other genes encoding for intracellular ADA. Intracellular, this is not extracellular, this is intracellular ADA. That is also forming tipless mounts, but this is still the results are not confirmatory because this is still, we assume that this is happening. We have to do additional tests to confirm that this is the case. We obtained further, we obtained several classes of mutants. All of them, these are knockouts. These are knockouts involved in other deaminases. So this should also generate ammonia. And if you have a knockout here, they also manifest in the mound arrest, but the mound arrest is phenotype is not as pronounced as in ADGF, the extracellular. They show a partial mound arrest. So we took the AlphaFold, which is the online software, and we modeled the structure. It is very similar to the human, this is called cat-eyed syndrome. The human ADA2 is very similar to the ADGF of dictyostelium. So dictyostelium can also be used as a model for cat-eyed syndrome, studying cat-eyed syndrome. This is the dictyostelium ADGF, this is the human ADGF, this is the overlapped structure of, the structures are aligning well. And the structures are aligning well, then we wanted to check what is ADGA, human ADA interacting with. So human ADA is known to interact with dipeptyl peptidase. Human ADA is known to interact. So the equivalent of dipeptyl peptidase in dictyostelium is called APRA. We obtained the mutant, and when we abolished the function of adenosine deaminase, we found no phenotype here. So maybe this is the phenotype may be apparent in the growth stage, but not at the developmental stage. So this is the dipeptyl peptidase. It is strongly, there is a strong interaction of dipeptyl peptidase. That is dipeptyl peptidase is the APRA with the ADGF, and this is the DHKD, which I said this is involved in the histidine kinase, which is involved in the pathway. So now, when we looked at the online database of human gastrula or mouse gastrula, during gastrulation, the organizer development happens. During early gastrulation, organizer. So the adenosine deaminase expression is stronger in both these embryonic stages, and so this. But when we looked at literature, not much is known about adenosine or ammonia formation during early gastrulation. So the very simplified model of how this works is like this. Cyclic AMP is the first messenger as well as the second messenger. Cyclic AMP is thrown out of the cell, it is secreted. This cyclic AMP is the adenosine is derived from cyclic AMP. From adenosine, ammonia is formed. So ammonia may be coming from protein catabolism, enzymatic reaction, adenosine deamination, several reactions. Even if you have a block in one, it is manifesting in the mound arrest. So this is the mound arrest means what? There is no organizer formation, there is no tip formation. So the low adenosine and high ammonia levels, that is responsible for genes that induces adenyl cyclase expression, that makes more cyclic AMP. The cyclic AMP relay is proper, then cyclic AMP wave is happening, as a consequence of which you have a proper tip development or organizer development. So I thank my guru for introducing me to this great organism dictyostelium, and also thank Professor Nanjundaya for introducing me to another great professor, Professor John Bonner. And this work was entirely done by Pavni Hathi, and you see these guys are judges, and they are all biased like me, and this is dictyostelium which is walking here, and all of them give I think give a score of 10 out of 10, and if you have time we can go on to the second part. I need glass cut.
Sir, I have a small question. So I'm still not completely clear about, can you just explain the role of ammonia in the formation of the tip in the tip development?
Ammonia induces cyclic AMP.
Okay.
And cyclic AMP triggers cell movement. So cell movement is necessary. And if there's a block in ammonia, whatever cyclic AMP is, not enough cyclic AMP is being made. And cell movement, collective cell migration, collective cell movement, this is also happening during gastrulation. And that collective movement is blocked, as a consequence of which tip is not formed.
Okay.
So ammonia comes first, then cyclic AMP. Then cyclic AMP triggers cell movement. That is what is happening.
Okay, so for this tip to form, is some sort of a differential concentration of ammonia needed over the cells, or is it?
That part we do not know. Okay. Differential but what we will think is ammonia is being made throughout. And there is something called slime sheet. If this is a slug, there is something covering on top of it, exactly like my hand. The front end is exposed, so there is a lot of volatile ammonia that is getting released, whereas the back, the diffusion is limited.
Right.
So that is a gradient. But there is what we do not know is what is the role of ammonia in the surrounding environments. because even bacteria, the nearby bacteria, dying bacteria, they can also rescue this phenotype.
Mm-hmm. Thank you.
So when we started with this, so initially when we started, I started with this model organism called polyspondylium. This is a fantastic model to work with, but it's very notorious because this forms symmetry breaking. Symmetry breaking, if you want to investigate, for example, here you see a round structure here. The round multicellular structure within a span of 40 minutes, it breaks its symmetry and forms a spheroid. From spheroidal symmetry, it's like spokes on a wheel, it forms a structure like this, so from globular to spherical symmetry. So, but this organism is very notorious for transformation. So if we cannot do transformation and do gene manipulation, then it's very difficult. But this, and also this model organism is very interesting because it is not making cyclic AMP as a chemoattractant. Instead, it is making a modified dipeptide called glurine, glutamic acid and ornithine, as a signaling molecule. So initially when we started, we looked at the role of caffeine in this problem. That is how, that led me to working on adenosine deaminase. This is a slug. If you transplant this slug and put it onto a plate containing caffeine, then what you have is multiple heads that are getting formed, multiple tips that are formed. Normally you have only one tip, that is at the front end. But when you put it here, then you get multiple tips formed. This is exactly a very nice problem to look at lateral inhibition. So high levels of adenosine, what is caffeine is an antagonist of adenosine. High levels of adenosine at the slug front is not allowing one more tip that is going to be formed along the slug axis. When the adenosine activity is blocked, then you have multiple tips that are formed. This is the typical form of lateral inhibition that you can see here. Sometimes you really wish you could do this. So I'll skip this for the sake of time. So this is a typical problem to look at. This is a fantastic problem from globular to spheroidal symmetry. Nothing happened. It's didn't work for us. So in coming to Arabidopsis, this is a model plant. So I'm going to. So this is the X-ray image of a nice Arabidopsis flower. What we do is there are 4 parts of a flower. This is the sepal, petal. This is the male part, and this is the female part. Upon fertilization, you get a lot of seeds here. And so here we try to estimate spontaneous somatic mutation rates in Arabidopsis. Spontaneous somatic mutation rates, even with sophisticated sequencing, it's not possible to estimate these classes of mutations because these events are very rare, happen somewhere in the structure, somewhere in the plant. So how we detect this somatic mutation is very simple. So you transform the plant with a marker. This marker, when it is expressed, the whole plant will be blue in color. The whole plant will be blue in color, but we deliberately make a mutation. In this, if you deliberately make a mutation, you can make a mutation precisely and introduce this back into the plant. Now, if this goes to a reversion process, that is, a mutated gene goes to wild type, then you see a blue spot here. So based on the number of blue spots to the total number of seedlings that you screen, then you can estimate a rough estimate of what are the mutation rates. So here we looked at these kind of vectors. For example, here this gene is mutated in such a way that it carries point mutation. So you introduce a stop codon there. If the stop codon is there, then the message is going to be truncated. The truncated message, it does not make any sense. So as a consequence of which you will not see. Suddenly, if it goes through a reversion process, then you will see upon staining, you will see a spot like this. Similarly, here you have a microsatellite. The same, for example, here you have stretches of G's introduced. And for example, if you have a stretch of 8 G's introduced here, either by addition of 1 more G or deletion of 2 more G's, the frame can be restored. So that way the frame can be restored. This is a microsatellite introduced. So once again the reversion. Here is a transposon in between a promoter, global promoter, followed by a reporter gene. There is a transposon sitting here. If the transposon jumps out, then the gus is going to be formed. Then you are going to see a blue color. And the last one is how we estimate homologous recombination. Here you have a G of, the gene is called the gus. If you can look at my hand, it is truncated and then introduced like this. If it goes through a process called homologous recombination, then you have the reversion, then the reversion can be visualized here. That is one problem that we are looking at. What we are looking at is how is hybridization, because Arabidopsis comes from a different source, how is hybridization influencing the spontaneous somatic mutation rates? So we took Arabidopsis from different parts of the globe, all of them you can collect, you can order it from the stock center. We made certain number of hybrids, generated stocks, analyzed the mutation, and also this is published now. So hybridization, we asked whether hybridization triggers one class of mutations over the others. But in all cases we found out that the parental sex makes a huge influence on the mutation rates. Then the second that we are looking at three problems in plants. One is how hybridization is influencing the mutation rates, how parental age at reproduction is influencing the mutation rates, how seed age is influencing the mutation rates. Because seeds are stored for a very long stretches of time, and if you store it for a very long time, the reactive oxygen species levels also are expected, it is published. Their levels are also known to go up. So do they affect the spontaneous mutation rate? The second one is how we estimate meiotic recombination rates. The assay is very simple. If you can focus your attention only on this set of chromosomes, these are called meiotic recombination detector line. So this is a homologous chromosome. In this chromosome you have a RFP, red fluorescent protein, that is driven by a seed specific promoter. So that, what do you mean by that? If this RFP is expressed, you are going to visualize this in the seeds. And this is GFP, this is green fluorescent protein. This green fluorescent protein, you are going to visualize the seeds. Suppose if there is a crossover, there is a single crossover here, only one crossover, then you are going to visualize, they are going to get separated like this, which you can visualize here. Then, for example, this green here, that you visualize the same set of seeds under RFP filter and a GFP filter. For example, if you look at green, this green is not reflected here. This is R. That R is not reflected in this portion here. So based on this, based on the fraction of red only and green only, you can estimate the meiotic recombination. But this line is not perfect. This has got pitfalls because the length, if there is a double recombination happening between these two markers, then it's very difficult to visualize. There is no way you can distinguish. So we are having markers that are very close by to each other and working on the effect of seed age as well as the role of plant hormones in meiotic recombinations. We found that strigolactone is an hormone that plays a significant role in meiotic recombination. Stop. So I'll stop here. Thank you.
Questions? Yeah.
I have two questions. One is, in that experiment where you added bacteria to slime mold plates.
Bacteria are independent. They are not physically touching.
Beg your pardon?
They are not physically touching the slime molds.
Okay, so you answered my question. I was wondering whether they reverted and started feeding.
No. They compartmentally, one compartment you have bacteria, but the bacteria takes time to die. So the recovery,
I get it. Yeah. Thanks. The other question was, quantitatively, how much of the ammonia produced is from cyclic AMP and how much via protein degradation?
We don't have an answer for this. Good question, but we don't have any answer at all.
Further questions? Yeah, Maitri.
Is there anything qualitatively different when, do ammonia and cyclic AMP change? I'm just wondering if the phenotypes of your adenosine deaminase and not having cyclic AMP have identical phenotypes.
Cyclic AMP, what you are saying is if there is a block in cyclic AMP production, will it give a similar phenotype? The answer is no, because cyclic AMP is involved early in development. So that means very early in development, even chemotaxis will not take place.
But so as morphogens, cyclic AMP and ammonia,
they interact because more ammonia is being made, it triggers adenyl cyclase expression, which is responsible for cyclic AMP production. Yeah.
So we saw that adenosine and ammonia both are kind of affecting how this will happen, like the tip formation will happen. So is it some kind of the ratio of adenosine and ammonia that is guiding
That
this?
is what initially we thought, but that doesn't seem to be the case, because when you have when you add ammonia, in spite of high levels of adenosine sitting in the plate, ammonia is able to rescue. Even at smaller concentration, even bacteria is able to rescue. So adenosine, the role is not very clear. I mean, it's very controversial. I mean, literature also sometimes they say yes, sometimes there is some other group which says no. Adenosine, it's not, whereas ammonia we can categorically say. The ratio I do not know. Is it the ratio of adenosine to ammonia?
Adenosine independently cannot do, like cannot
That
rescue.
is what, if you add adenosine to wild type, okay, then it should induce the Mounderus phenotype. That does not happen.
Okay. Yeah.
Wave patterns are different for wild type and the mutant?
Yes, I mean, this is a hypothesis. In the mutant it is circular, whereas there is a symmetry breaking which is happening. That symmetry break, from initially it starts with circular waves, and then it becomes a spiral. Then in the mutant it stays as a circular wave. It does not break. I do not know the reason for it.
Could it be because of the wavelength of the waves? I mean, basically you have a much larger spacing between successive waves in mutants. Have you tried measuring it?
No.
Okay. Because if the refractory period, excitable medium. Yeah, so basically they have to, so for example in excitable media models, by altering the refractory period you can actually make spiral breaking, breaking into spiral waves very hard in Fujinagumo and so on. Yeah. So I'm saying that in Fujinagumo, for example, breaking a circular wave into spiral becomes very hard if you make the refractory period, alter the refractory period. So maybe something like that might be happening here. So you could just try measuring the spacing between successive waves and see if that changes. Yeah. Any other questions? So I'm just wondering, suppose, once the dictee have started sending out the cyclic AMP, if at that point you suddenly add again a lot of bacteria, can it stop the cyclic AMP signal and revert back to feeding the bacteria, or is it like a irreversible thing? Once they
think
decide
beyond
to
the slug stage you cannot do it, but the aggregation stage I do not know.
I see. Okay.
Slug stage I think they ignore.
All right. Okay, so if there are no further questions, let's thank the speaker once more. And we now break for lunch. And please be back here at 2 for the next talk.